Good morning, everyone, and welcome to today's webinar on smart analysis of butter using that smart SpectraStar XT NIR Analyzer. As we look forward to National Butter Day, we are excited to show you how this advanced NIR technology makes butter analysis faster, easier, and more precise, measuring key parameters like moisture, fat, protein, and solids, and any other more in just seconds without any chemical or waste of your substances. So, today's session will be led by my colleague, Mr. Gaurav Thomre. He's an application chemist at Encrom Enterprises who has strong expertise in NIR instrument and their applications. So, feel free to drop your questions anytime in the comment section. We will take them during the session or in the Q&A at the end. Okay, and I think more participants are joining. So we will start in within 2 minutes. So with that, we'll begin and over to you, Gaurav.
Thank you so much, sir.
Yeah.
So, dear attendees, I welcome you all to this engaging webinar over here, wherein we'll see how this Unity Scientific SpectraStar XT RNIR would be helping you with optimizing your butter production. Like how this Unity Scientific SpectraStar XTR, like what is it? How will it be useful to you? And how means in what all aspects it would turn out to be useful to you? What are benefits would you get out of it? And also we'll see through like what is this whole technique about. Okay, so what this NIR and butter will do. Mm, yeah. So talk like our star of the day, like butter. So let us understand like what is butter and why do we need to analyze butter and how butter is overall evaluated for its quality and like most importantly we'll see like the how this technique would turn out to be helpful to the industry because industry are the max producers of this kind of substances like butter. So, like today we will see how this SpectraStar XTR can help the butter producers. So, like this SpectraStar XTR NIR, this is a NIR technique, near infrared spectroscopy. So, this being a secondary technique, how it can turn out to be helpful to the producers by analyzing their maximum samples and for when it analyzes those varied samples for them it would give you best results how the results that you get and to achieve the batch to batch consistency for each batch that you produce for your product that you have. It could be butter of different flavors of different class and even other dairy products. And overall, how can you attain the batch to batch consistency and hence make profits by saving time, chemicals, and other resources? So, butter. This butter is nothing but a concentrated dairy product, which mostly has about fat and protein in it. So this product is being made from milk. So milk contains some amount of moisture, some amount of water, some amount of other fatty acid and things. But then those things are eliminated out of the milk, and then during the processing steps, only 80 to 85% of the milk fat is being considered and using that portion of the milk this butter is being produced. Okay, so let's see how this butter is being pressed. So here if you see, so they initially the making of butter starts with collection of milk. So this milk is collected from farms. It could be a dairy center wherein all the different milks are being collected from different and then it is mixed and homogenized at a dairy center, and from the dairy it is then um, sent to the factory where the product butter is made. Okay, so there into that factory, this milk is being agitated to get its cream separated from it. So this cream is the portion of the milk that we are interested into, and through this cream we will be preparing our butter. So this cream gets separated from the milk, and after separation of the cream, the leftover liquid is being out. So the cream that was separated from the milk, it so as to eliminate the bacteria and other microbial organisms that grow during the process of vegetation, the cream is being pasteurized. So when it we all know the process of pasteurizing, so it helps in killing the microbes and other organisms that grow in any culture or in milk culture mostly, I would say. Okay, so it we do that by subjecting any kind of substance to a very extremely high temperature. So after subjecting that substance to a higher temperature, we suddenly cool it. So when you cool it, the bacteria or any organism that is there present in it or is trying to flourish in that environment doesn't get any chance to flourish when you suddenly cool it. Okay, so you let this cream to cool. Then once you cool it, you start churning it. So churning is the process in which what happens is the cream which is cooled, the cooled cream, it still has some amount of water or some amount of moisture in it. So while churning, what happens is the fat globules that are there in the cream, they get accumulated. And once they get accumulated, the cream we start to churn, it is at some room temperature. So again, the water starts separating from it. So we need to wash all the lactose containing and other matrices that are there other than fat and protein from our cream. So it requires wash. So you need to wash your intermediate butter, I would call it here. Okay, so you wash your cream here. So once you wash it, we subject our intermediate butter to salt. So salting again, we are very well aware about the process of preservation. So salting is one kind of a preservation step that we follow. So wherein what happens is the salting. What actually happens during salting is you add salt, and you what you do is you concentrate the culture that is there. So when you have a highly concentrated culture, you don't have any space for the there is no space, I mean, for the microbes and other organisms to grow. Okay, so thus this increases the shelf life of the butter and even give it a taste that we like it for, right? So this is how, I mean, the salting is a very important step here in butter. And again, the salting process, it varies like depending on the type of butter that you're producing. So once you salt your product butter, and then you need to keep it for cold storage. So when you salt it, the mixing of the salt has to be done to homogeneously mix the, I mean, distribute the taste of salt throughout the butter, making it taste evenly consistent and better every time you have it. And then once you subject it to salting, it needs to be stored in a cold temperature. As we all know, at room temperature, the butter melts off, and then the containers or any package that is holding the butter would no longer be able to hold it. And then once you store it, in the cold temperatures, it is ready for distribution and it's further processing. Okay, so this was just an overview. So we'll see like how the detailed production of butter happens. So firstly, like I told you, the fresh milk is collected from the farms, and it undergoes some kind of testing. So yes, of course, the milk has to be tested for different parameters that it is evaluated for. Mostly, the milk is evaluated for the amount of moisture in it, the fat content it has, and also the protein content. Because again, milk being a raw material for various types of dairy products, so cheese has been made out of it, milk powders, this butter, then buttermilks, and many more things are being made out of it. So the fat content and the protein content has to be checked, monitored properly. Okay. Again, so when you test it, it ensures the only the high quality milk is being supplied to the producers. So eventually, we get a higher quality of butter that's produced from that milk. So once the milk reaches to the factory, what happens is the cream separator, well, like I just told you, the cream separator, the milk is being poured into huge containers. Those are centrifugal separators. So what these separators do is they agitate the milk inside it, and they separate the liquid and the cream part. That cream part indicates the when you agitate a milk. So what happens is the fat gets collected at the top of the vessel or container that you are agitating it into, and then the liquid which has lactose, the other moisture and water, hydrophilic substances are there in the liquid which is left behind. Okay, so that cream is being separated. Once it is separated, concentrated. So mm, yeah. So the cream that is being separated, like I just told you, it has to be pasteurized. So when you pasteurize the cream, so during the agitation, any microbes or any organisms that are being formed, those need to be destroyed so as to increase, so as to get a better butter. And even it should be devoid of any kind of microbes or those things. As if our cream has been initially infested by these microbes or other organisms, the taste would be affected, its quality will be affected, and most importantly, the shelf life. Though you prepare butter with these microbes and things inside it, but it won't be staying there for a longer time in your fridge, I would say. है ना? So it needs to be pasteurized. So in this pasteurizing process, the cream is being heated at in this range of 85 to 90 degrees Celsius for 15 to 20 seconds. Immediately, it has been cooled at the, I mean, is being heated to this huge temperature, killing those microbes and other organisms in it. Right after cooling it to temperature, it is suddenly cooled and stored in these vessels. So when it is stored in those vessels, the butter also, I mean the cream, it also undergoes a step known as aging. Like we all must be aware, like every dairy product that we consume, uh, once it gets aged to some period of time, you get that desire, I mean the flavor that it is known for. So suppose if we talk about cheese, so again in cheese we have various different kinds of cheeses available. So the texture, the taste that we experience for each different kind of cheese that we've heard of, so it could be mozzarella cheese, you have cheddar cheese, you have other spreadable cheeses, and many such kind of cheeses. So we know those cheese because of their taste, their texture, and the flavor most importantly. So all these flavors and the textures are being achieved by this step of aging that it follows during the process of production. Okay. So once you cool it into these vessels, the again what happens is when you cool it and when you store it, the water inside the cream again starts separating. So you start churning the cream that is there in the vessels. So once you churn it, what happens is the water again stored in small air bubbles, or water means small gaps are being created once you agitate something and store it, right? So into the small air pockets, water is being stored. So when you churn it, what happens is the water inside the uh, intermediate butter, I would say. It is left out of those is squeezed out, I would say, is squeezed out of those intermediate butter. And then again, the butter or the cream is devoid of maximum moisture, means the during the step of churning, what happens is maximum moisture from the butter is being eliminated out. So the liquid that is being drained out from the cream while churning in during the soft churning, the buttermilk is separated. So this buttermilk is nothing but the liquid part that is separated from the cream while churning. So it has some amount of fat in it, it has some amount of protein in it, but most importantly, it has that buttery taste, you see, that we like our buttermilk for. So here in India, mostly we have spiced buttermilks, right? छाछ जिसे हम कहते हैं. So again, to this buttermilk, the raw buttermilk, which is unflavored and doesn't has any salt in it, or it is same as some buttery water that you are drinking. So to that tasteless buttery water, different spices and herbs are being added, so so as to we get that chas thing that we like it for, right? So that buttermilk has to be drained from the cream so as to get a better butter, and also at the right time, the good quality buttermilk has to be separated from the cream. Okay, so that is what is done here during the step of churning. So if you see here, the cream has been whipped. I mean, here you could consider it to be the step of churning. So when you churn it, the fat globules, they compile to each other and forming bigger fat globules. Like, also what happens is the fat globules which are in bigger size, they get reduced to a smaller size, and also you get a uniform particle size, I mean the fat globule size that you achieve during the step of churning. So that ensures the taste of butter that you get, and also the texture that you achieve once the butter is prepared. Okay, so after those globules are accumulated, the milk buttermilk gets separated. So once the buttermilk is separated, you use this cheesecloth. So this cheesecloth kind of acts as a filtering system, so wherein you just put in your cream, which has some amount of again some amount of moisture left in it. So you squeeze it so that the excess of buttermilk or any water in it gets eliminated out. So the thing which is left back in that cheesecloth or any filtering film that you use it for, so that is what is used for further processing of the butter. So like I told you, washing and working. So this washing and working is a mm, even a better step than churning, I would say, wherein what happens is the cream, which is the semi-solid cream, which is left back in the cloth or the film, is then washed with cold water. So here the cold water is used for washing the fats and the lactose containing liquid, because what happens is if you use hot water, the flat globules and other things in the cream, semi-solid cream that is left back, so those would get washed off, and you would lose the yield of butter that you would get at the end. So make sure your means this cold butter has to be used there for washing. So when you wash it, excess of these lactose and other non-required components that are there in our intermediate butter, they get washed off. Okay, so as it is devoid of any moisture in it, any other components than our desired components, it eventually increases its shelf life, its texture, and its quality though. Okay. So once you wash it, then the cream is then subjected to salting. So now I won't call it as butter. This would be our unsalted butter, a type of butter that is again available in our markets. Okay. So here is where you get your unsalted butter, and means the step until which the salt is not to be added to your cream. It is an unsalted butter. Okay. So this unsalted butter can now here this unsalted butter can be packed and could be sold out. again, before selling out, it needs to be treated cold there so that it attains its shape. And then once it attains that shape, it can be transported ahead. But then mostly we like the salted butter, right? So the salting process is being done over here. So this salting process is a process, like I told you, it is in this process you give flavor to your butter, and even by giving flavor, you also add a kind of a preservative to it, so which preserves the butter and increasing its life. So, like I told you, when you add salt to any kind of a culture, so it concentrates it. So salt, what it has this property of, it absorbs any moisture that comes in contact with it. Okay, so it absorbs that moisture, and what it does is it creates a dry kind of an environment. Yes, it has that velvety, fatty texture, but it is dry. It is devoid of any moisture in it. Okay, so the salting process ensures the taste of the butter and the overall shelf life of the butter once it is processed. So once you salt it, salting it, the butter needs to be packed. So based on the different types of butters that we have, different packing materials are being used. Okay. So for spreadable butters, what we follow is, so for spreadable butters, what we follow is we pack them in those rectangular packets, right? So that you can easily collect the butter from the container and then apply it over your foods, right? So again, you mostly we have those butters coming in the for those rectangular cuboidal shapes, right? So based on the butters that we means that is being produced, different packagings are followed. So once it is being packed, it is stored in the cold storage so that it attains the shape of the vessel or the packaging material into which it is wrapped around, and then it is distributed. Okay. Once it is distributed, we consume it. So where would the NR fit in? Like, how will the NR be useful here in this whole process of butter production? So, like I told you, while means how do you evaluate your butter? First of all, tell me that. So, when you evaluate your butter, so see, we have this poll coming up which reads as how do you evaluate your butter and other dairy products? So, dear attendees, I request you all to answer this poll so that we could have a better discussion ahead with respect to our butter analysis and how this NIR could be helpful in your butter analysis. So we have this option of wet chemistry methods. So if you all use the primary methods for your water analysis, you could opt for this option. Next is outsourced testing. So when you give your products to some third party or some other testing organization and get them tested, so you could opt for that option. Or if you use NIR, near infrared spectroscopy, any sort of NIR that you label at your end. If you are using that kind of NIR, so means you can opt for it. And if you are using some other instruments, you could specify those. So once you answer the poll, just anybody from the attendees could put a thumbs up or anything, or give me an intimation in the chat box so that we could proceed with our webinar for ahead and see how this NIR could be helpful to you. So, dear attendees, let us see like why NIR should be used. And first of all, let us see like is it really helpful? Oh, yes, we have answers to the poll. So, like, and can see 20% of our attendees are using this wet chemistry of an kind of analysis for analyzing their dairy products and butter samples. And I find a huge amount of attendees using NIRs of different brands. So let us see how the RNIR, the SpectraStar XT from Unity Scientific, could be helpful to you. So when you're analyzing different parameters like moisture, sugars, proteins, fibers, ash, fat, and oil, and other parameters. It consumes your time, your chemicals, and your energy. So time by the time it takes for the sample analysis, chemicals in the form of the reagents and those things that you prepare, and energy for preparing those chemicals, and even in the human efforts that are required for preparing the chemicals and doing the whole analysis and getting the results out. So there's a huge investment that is required for getting the results out and for maintaining batch to batch consistency. Why? Because you follow all these primary methods of analysis. So for moisture, you subject your samples to oven dry. For ashing, you keep them for muffle furnace. For evaluating fat, you subject them for Soxhlet kind of extraction. For evaluating protein, you keep it for Geldal extraction. For evaluating sugars, you keep it for iodometric titration or HPLC. And fibers, like we know, those are evaluated by acid digestion and then ashing. So, NIR is something that would help you with. Okay, so we have another poll over here, which is asking like, what is your biggest challenge in butter quality testing? So, the options that you have are long analysis time, high cost of reagents and chemicals that are involved in your analysis, inconsistent results. Yes, based on different analysts, I think inconsistent results should be achieved, or could be even because of those sample which was being you give analyzed there. So next is we have labor intensive process actually. So testing means if we consider this to be from the producer's point of view. So yes, testing is a labor intensive process and need for higher throughput. Yeah, of course, means some company who's making good on production. Now, if they wish to upscale their analysis, they can go for it, right? So, dear attendees, mm, y'all should even answer to this poll based on the based on your choice, so that we'll see like how this NR would be helpful to you. Yeah. So I see most of my attendees, they are worried about the high cost of reagents and chemicals that are involved in the analysis, and also they are striving with the inconsistent results that they get from their primary methods, or with what any method that is used for their and evaluating their butter and dairy products. So why NIR? Because NIR, here in short, I would tell you that it would help you save your time, it would help you save your chemicals, and it would help you save your energy as well. Why? Because NIR, being a secondary technique, like I told you, it ensures that what happens is the main difference between the primary method and secondary method here is the speed of analysis that you get. Okay, the speed of analysis plus the sample throughput. So we would see like how the see how our SpectraStar NIR works. But before that, let me give you an overview, like this technique means, what it does, and how it works. Okay, so let us see that. So this NIR, so NIR, like I just told you, it's abbreviates for near infrared spectroscopy. So this near infrared spectroscopy or near infrared region comes from our electromagnetic spectrum. So in our electromagnetic spectrum, we know there are different regions. So this NIR region is the region right before the IR region. Okay. So from this IR region, this NIR spectroscopy uses this near infrared region, which ranges from means we could consider this to be a very general range wherein it starts from 700 nanometers to 2500 nanometers. In this range, why this NIR range is only selected for evaluating different parameters from your sample? It because it has the um, best attributes. Means in this spectrum, what happens is these set of wavelengths, they has the best attributes for analyzing different sample types. So like it could analyze your solids, your liquids, your other kinds of samples. So all these different kinds of samples are only available in our one matrice, that is our food. Okay, so different kind of food samples could be analyzed using this NIR. So in this NIR, like how this technique works. So the range that I just told you, from 700 nanometers to 2500 nanometers, to this range, for means for producing this range, how what would ensure that this range comes out from any lamp source that you are having. So what happens is, the from the lamp source, the rays are made incident over this grating. So this grating, what it does is it ensures the full wavelength range, whatever your NIR offers. So through this grating, the monochromatic grating, the selected wave, I mean, all those wavelengths are made incident over your sample one by one. And then when your sample gets exposed to those radiations, what happens is it kind of interacts with it. So the interaction kind of happens in the form of what kind of interaction happens when light interacts with any substance? Mostly it is reflection. And in some case, when you have some clear or transparent or translucent sample, you have your transflection happen, right? So that is what happens. So during this process, the NIR, what it does is it does rapid analysis. So the advantages of NIR, like why an NIR should be used, why the secondary technique has to be used over your primary method, because it gives you rapid analysis. So when you have your multiple batches being prepared of any sample that you are producing. Okay, so it requires decision making, right? So any sample that is into your specific limits, or it is not, quick decisions has to be taken so as your production line doesn't stop, and you don't face any losses because of those production, I mean stopped production line. So it provides rapid analysis of your various sample. So compared to a traditional analysis method, it requires no sample preparation, no chemicals, and no consumables. So any product that you are analyzing over the NIR, shortly you would see that it doesn't require any sample preparation. Barely it would require any simple sample preparation, wherein what happens is, suppose if you have some hard sample, some mm, a hard kind of a solid sample, it just needs to be crushed, it needs to be homogenized. And once you homogenize it, simply it needs to be stored, it need to be kept in the sample cup and then get it analyzed. Again, when you are crushing it, it doesn't require any chemical. Also, the NIR also doesn't require any chemicals for its functioning or for the analysis of sample. Okay. Next, another best thing about this NIR that it performs non-destructive kind of analysis. So what happens is, when only the light interaction happens with your sample and nothing else. Again, the light, it only interacts in the form of reflection and transmittance to your sample. So it doesn't destroy a sample. So this NIR, it gives you your sample unharmed, and you could again use that same sample for some other kind of testing that you do. Okay. So when it does this rapid analysis and doesn't use any chemicals and doesn't need any sample preparation, and even when it's doing this non-destructive kind of analysis, let us see like, how this NIR works. So what happens is the near infrared radiations you would see here. So these near infrared radiations from the lamp source are made incident over your sample, and then the reflected rays or the modified rays which have interacted with your sample are then captured by the detectors. Now these detectors only would ensure like what kind of interaction has happened with your sample. So is it a kind of a reflection that has happened or some kind of reflectance that has happened? So based on the kind of interaction, the detectors, what they do is they collect that information, and that information is then collated with the kind of algorithm using which the calibrations have been prepared. So this calibration is nothing but a set of primary data that you feed in the NIR instrument. So this primary data is the method that you follow for your analysis. So in the initial slides, like we just saw, for evaluating fat, you subject your sample for oxalate extraction. So from that primary method, you would collect the data, feed it into your secondary instrument, and then that secondary instrument, what it would do is using that primary data, it would analyze your unknown samples, and based on the primary data's information, it would give you results for your new samples. How? By some non-destructive kind of analysis. Whereas your primary method, it completely destroyed your sample. It consumed your time. It also consumed your sample too. Whereas the secondary method would only consume some amount of your time, and that won't be more than 60 seconds. Okay. So this is how NIR proves superior over the primary methods, and also saves upon your time, your chemicals, your energy, and other resources. So in this range of NIR, we have three modules. So we have our SpectraStar XT3. So this is an entry level analyzer. So it analyzes any three parameters from your sample. So it could be your protein, ash, and moisture, or any other parameters based on the product that you are analyzing. Next, we have this SpectraStar XTF. So this SpectraStar XTF is for the grains that grains and the floor that is prepared from those grains. So it is able to evaluate that. And next we have this SpectraStar XTR. So this SpectraStar XTR is our ultimate module in this product range. So this is able to analyze every sample right from your grains to your finished products to your intermediates, and even your complex samples of various natures. Okay. So let's see like what these instruments have them in common and what is their actual difference. So there is not much difference here physically. If you see all these three instruments, they look identical, but the difference lies here in their make. So here, if you see the SpectraStar XT3, it works in this range of 1400 nanometers to 2600 nanometers. Okay. In this range, it only measures the reflectance that is being produced or captured by the detector. Okay, so any sample that you analyze, only its reflectance would be captured there. Okay. So, from within this range, when it captures only the reflectance, it only gives you the three parameters. So it is programmed in that manner that it would only give you results for any three parameters. Next is we have our SpectraStar XTF. So this XTF again have this same range, but the make is made in such a manner that though it is again capturing the mode of measurement is reflectance here, but is able to give you other parameters as well apart from the basic three parameters in that product. Okay, so next we have on our star, that is SpectraStar XTR. So this NIR, from the heading itself you can make out that it is suitable for food samples that we consume, feed samples that are animals or browsing animals they consume, even forage samples that we give them for their health benefits. Okay, so all these samples could be analyzed in this range. So to analyze all these samples of different nature, it works in this range of 680 nanometers to 2600 nanometers. This is a huge range wherein it analyzes n number of parameters from your sample. Again, all these NIRs that we have just give you results within 60 seconds max with no sample preparation, no chemicals to consume, and very less energy is also again consumed by the instrument that we have. Okay, so I see Mr. Shankar Mishra has raised his hand. So, sir, I would answer your question after a while. Just let me complete this, and then we'll start with the Q&A session. Okay. So all these NIRs that we have here, if we just see the detectors that we have, so for measuring the reagent. Hello.
Please continue, Gaurav.
Yeah, thank you, ma'am.
Itu iba
So for
on that because it was carried by the
yeah. So for analyzing various samples and for getting those results from it, it has this high performance ultra cooled in-gas detector, extended range detector. So this in-gas is the are the different elements that are used in this uh, detector that we have. So it indicates indium, gallium, and arsenic. Okay, so instruments that we use. So this the SpectraStar XT3 and XTF have the same kind of detectors, but the internal make is different. Whereas our SpectraStar XTR has this dual performance high industrial grade um, detector which is able to measure the reflectance as well as transflectance from different samples. So for measuring the reflectance and transflectance, the rays has to be made incident over the sample. Okay, for that the lamp source that we have is the tungsten halogen lamp with this mean time between failure rating of 10,000 hours. Okay, so for 10,000 hours, this lamp is functional once you install it. Okay, and again, best part about our lamp is again that it is user changeable. So you don't require the service person to come up every time to perform any wear and tear of your I mean your instrument. Okay, so all our instrument that we have are completely sealed. They don't have any fans or vents in it. So as our instruments are completely sealed, they are made to withstand the factory flows. So on the factory flows, you see there are huge vibrations and dust flowing everywhere. Okay, so our instrument is all unaffected by it, and as our instrument is unaffected, excuse, the data that we get is again unaffected by all such other metrics that are there in the environment where it get tested. So our instrument, that is SpectraStar XTR, is a very easy to use instrument. It is a standalone kind of a design, simply plug and play solution for your laboratory and netline productions. So it could be helpful to you for your QC analysis, wherein you check for your final product, and in the QC, or you also evaluate your raw products, and it the production line, you could check for the intermediates that are being formed. So while the process of production is happening, you could check for the intermediates for their quality, their consistency, and for other parameters over which you wish to evaluate those. So during the analysis of different samples at your final product or your incoming raw ingredients or your intermediates, these samples could be of various natures. So our SpectraStar XT analyzer is able to handle various kind of samples of solid, liquid, powders, seeds, and other kinds of nature. Okay, from all these different kinds of samples, it is able to analyze various kinds of matrices. So, to name some, we have moisture, protein, fat, sugars, and fibers, and even some complex parameters like fatty acids, lignins, amino acids as well. So, to operate our instrument, we have this UScan software. Now, this UScan software, it comes in with the instrument itself. You don't need to purchase it separately. Okay, so this instrument is, this software is very easy to use software. Means the interface is such that within just 15 minutes, even a layman who doesn't has any scientific background or doesn't know any terminologies would be able to use this software and get user independent results. Now, a poll was being posted in its slides, if y'all remember, wherein the my attendees, y'all attendees, you said that you were striving with inconsistent results. So this um software that we have, so and along with software, the make that we have, our SpectraStar XTR, our XTR instrument is, our SpectraStar X NIR series, they are made in such a way that those are able to give you user independent results, because the sample prepare, you don't need to prepare samples. So just a simple processing is required, and once you keep your samples over the sampling window, you just need to scan them, and with those rugged calibration, get better results and user independent results, in respect of any analyst who's analyzing any sample. Okay, so from this software and through this instrument, you are able to get your data out. Okay, so normally you can export the data in the form of Excel, PDFs, or other manners, Word, and those things. And even you could generate reports for your different samples. But it also gives you a better hand wherein it has this LIMS compatibility. Now, this LIMS compatibility, this LIMS stands for Laboratory Information Management System. So herein what happens is any sample that is analyzed, so the uh, analyzers or the monitors, the different monitors and different department has different parameters to look for, right? So the QA person would be looking for something else, the QC person is looking for something else, and the production people would be looking for some other parameters. But they would have these parameters in common, but the values for these parameters would be different, right? So this LIMS compatibility, it ensures that the on the um department, all the data gets segregated and easily reaches to the person who's looking for any data from the whole compilation of results. Okay, so our SpectraStar XT NIRs could be helpful to you at all these steps. So like I told you, during the in process in incoming of raw ingredients. So the milk, here we would take our example of milk. So your liquid milk, when it comes to your factory, it should be evaluated. So mostly the moisture, protein, and the fat content of it is being analyzed. Okay, so you can use our SpectraStar XT NIRs for that purpose. Next is during the in-process monitoring. During the in-process monitoring, suppose when you've subjected your cream to some that separation, and when you separated and the buttermilk is being separated from it, the liquid is being drained out, so you could check for the extraction efficiency. So initially you showed some amount of fat in. Okay, so now when you've subjected the milk to the cream separation, so when the cream is now separated, so the cream is nothing but the fat content in it. So you could check if initially the fat content in our milk was XYZ. Now, after separating the cream from the whole milk, now you have some ABC kind of fat content in it. So you could compare the data at this on the same instrument at the same place. Next, from that cream, now that you've prepared the or if your butter is in process, like you have the intermediate butter before salting, or any step before packaging, so you could even check for its quality again. So at the same place, at the same instrument, you could check for all these different things and compare the data. So our USCAN software also allows you to compare the data. If you see here in this window, so it allows you to compare each parameter from different samples and even check for their profile. So you could compare the spectra for different samples that we're analyzing. So this is how our means at all these different levels, our instrument, our spectra, our XT NIRs could be helpful to you. So, by considering our substance or our simple butter. So from our butter, like we saw, milk was used. So just I told you that milk can be used, can be analyzed over our NIR for different parameters. Next, when the cream is separated from the milk, again that cream could be analyzed over our NIR. From the cream, the buttermilk was separated, so even that buttermilk can be analyzed. So what happens is, again, these creams, these pasteurized milks and buttermilks, they are subjected to further processing. So pasteurized milk, what happens separately in a different production house, what happens is the milk powders are being produced. Okay, so you could even check milk powder that is being produced from the milk that has come into the production house. Now from the cream, again, based on the different grades of creams, some high fat cream, low fat cream, or medium fat, or any whatever is the content specified over the label, the cream could be checked for it. Again, this milk or whey is again checked for the amount of protein that is being there in it. Okay, and I've even heard about some milk. I mean, this buttermilk powders. So again, those could be analyzed over our NIR. And finally, the final product, a product that is made, could be analyzed over our NIR again. So these are the different ready calibrations that are available with Unity Scientific, which can be used for analyzing your various samples. So we have our buttermilk powder, cheese, then creams, our yogurts, ice cream mixes, mayonnaise, our butter, and all such different kinds of samples could be analyzed using our SpectraStar XT NIRs. So, when you are evaluating all such things, your instrument needs to be calibrated, right? So, like, means on what basis do you ensure the quality and the authenticity of the results, right? So, you need some kind of calibration, right? So, yes, we have this pool coming up wherein which is asking for better butter analysis results. Do you need to follow tedious calibration process? Yes. Now, my attendees, please tell me, like, if at all you are using some kind of instrument for your analysis of different samples of different dairy products or your butter sample, does your instrument require calibration? And what kind of calibration does it require? First of all, tell me that. Is it tedious or not? Of course, every instrument requires calibration, right? So as to check if the instrument is working fine or not. So yes, mostly I see my attendees, they are saying like they require TS calibration processes for their for calibrating their instrument. So our SpectraStar XTR again proves superior to other techniques. Let us see how does it do that. So our SpectraStar XTR, I mean our SpectraStar XT NIRs, they come in this patented true alignment spectroscopy, wherein what happens is this true alignment spectroscopy, from the name itself, you can make out true alignment spectroscopy. Now, any components that are there inside the instrument's hardware, they need to be calibrated, they need to be aligned. Only if they are aligned in a proper manner, only then you would get those reproducible, reliable, and consistent results, right? So for calibrating the instrument, it comes in with this task standards. So these task standards are nothing but different components that are there in your instrument, which needs to be calibrated. Okay, so as our instrument uses a wavelength range, the photometric part of the instrument needs to be calibrated. Okay, photometric means it deals with light, so the light needs to be corrected. Next is the wavelength standard. So this wavelength standard, it would ensure that any wavelength that is coming out of the instrument or from the lamps, it is of the said wavelength. Oh, next, the dark standard. When you place your sample over the sampling window, it all gets dark inside the instrument. So that darkness again needs to be corrected. Oh, now all these alignments and all these things, when can they get hampered? It is when you move your instrument from some one location to another, or you kind of perform some kind of servicing to your instrument, right? So this true alignment spectroscopy is a very um newer concept of calibration, wherein what happens is, like this type of sample analysis, you could even calibrate your instrument with these three standards and calibrate your instrument to your factory specifications. Okay, so these standards that we have for the correction of the for light that enters our instrument, the wavelength and the darkness of our instrument, these three standards are NIST traceable. So NIST stands for National Institute of Standard Technology. Okay, so this ensures the standards that you have or the calibrants that you use for calibration of your instrument are really authentic, and whatever they claim to do, it is able to do that. Okay, so that ensures the reproducible, I'm not reproducible, but easy calibration of your instrument. So just load these calibrants over your sampling window and then start with your sample calibration. I mean your instrument calibration. So your instrument gets calibrated at your own end. So you don't require a service engineer uh, uh, from the source company to visit your vicinity every time, perform the calibration. Anytime you find any deviation in your results, or if you've subject your sample to some kind of servicing, or some kind of means, if you've changed the location of your instrument, just perform the spectroscopy, which is true alignment spectroscopy, and your instrument gets calibrated to that location, to that environment, and to the change that you've done into your instrument after some servicing. Okay, so next, after calibrating the instrument and analyzing all your samples and different things, like the data management. So the data management has to be done properly, right? So here our SpectraStar XT NIRs, they come in with this. Wow, we have this another poll wherein which asks about how do you like to monitor your analysis data. So mostly what happens is, analysts are the ones who are just analyzing the samples and checking for the results. Whereas the higher authorities, like the managers and those people over whom the whole production line and the those responsibilities are over their shoulders, they can't go to each of their production sites and check for the results, right? So yeah, like you like to monitor your analysis data. How do you like to that? Distantly or on site? So I see my attendees saying that they like to do it on site, but on site reaching to that location is a task, right? Then even need to check for the availability of the instrument so that you don't reach to that location at a wrong time and the simple analysis is going on, and therein you stand and you ask the analyst to show you the results, right? So that would waste the time of the analyst, the time of analysis, and the production light would also wait for some time till that particular sample gets analyzed. Okay, so as to overcome all of these things and to ensure consistency in the results and ease of the monitoring, we have this KPM link. So this KPM link, it is a kind of a feature that Unity Scientific offers, wherein what happens is you can monitor your data distantly. So any instrument that is connected over your KPM link, you would be able to see the data for that particular any sample which gets analyzed over it. So right means I would say means if your sample is analyzed at a particular location, at the other you'll be able to see the results for it. Okay, so basically you could even see for the results of it and even monitor the health of the instrument, which is very important. What happens is in a production line, if the analysts are analyzing their samples, so they won't check for the lamp bars and the what I would say, other factors that needs to be checked every time because they are busy analyzing their samples. But the person who's monitoring the system through KPM link would also be able to see the health of the instrument. If the instrument is working fine, if all the components in the instruments are in the said range, in the said limits, and those things. If your lamp is not overused, and those things could be monitored or distantly, and even decisions could be made distantly and based on your call, you could take decision for your samples and make best out of it using our KPM link feature from Unity Scientific. Wow, so next is what we have this UCal4 software. So let me tell you. So these are the ready prepared calibrations. So for when you are analyzing all such samples from this list, you do need to prepare the primary data and collect the spectrum and do all of those things. Okay, because you have ready calibrations for these samples and you could directly load these calibrations over your instrument and get your products analyzed. Okay, so apart from these samples, if you have some different product which is to be analyzed, you are interested in analyzing some what example can I give you? Mm, suppose we have some kind of fruit juice or so. Okay. Now these fruit juices, here we could consider as apple juice or so. So the which is not prepared synthetically, but actually the juice which is extracted from apple. So and again, now these apples that you are producing the juice from are some special kind of apples which are not in the world. Okay, you are working on some kind of a private project. Okay, so here you even develop your custom calibrations for your custom products. Okay, so now this UCal 4 software comes in picture. So this UCal 4 software, what it allows you to do is it helps you to collect the primary data from the prepare the custom calibrations for your new products or some novel product which is not there in the list of calibrations. Okay, so for that product, you could define your own desired parameters and your own samples and kind of products could be analyzed and could be evaluated for different parameters over our SpectraStar XT NIRs, but the custom calibration would be prepared using this UCal4 software. So this UCal4 software, what it does is it uses your primary data that you feed it from the primary methods. Okay, so here is where you define your product. So for a particular sample one, this was the data for the fat and oil content. We are not able to see this properly. So here I'll explain you. So suppose this is sample one. In sample one, the fat and protein content was defined here. Okay. So for sample one, you feed its spectral information here into the other window. So this UCal4 software does is it compiles this spectral information and this primary data to plot such kind of calibration. So now this calibration is nothing but the collection of data points that are used for analyzing your unknown samples. Okay, so when you are feeding in information about n number of samples, their primary information and its spectral data, it would compile both of these information, preparing this calibration. So now using this calibration, you are ready to use your NIR for your unknown samples. Okay, so any novel product could also be analyzed over our SpectraStar XTR with this UCal with a application prepared by this UCal4 software. Okay. Now, as we saw all these features, like it is able to analyze our samples within just 60 seconds, doesn't use any chemicals, it require any sample preparation step. It is even easy to calibrate. It doesn't require any fanning, venting, and able to withstand factory flaws, and it is able to view reproducible results and all these things. So, is it recommended or does it follow any kind of guidelines? So, yes, it follows this ISO 12099. So, this ISO 12099, it talks about this use of NIR spectroscopy in analysis of different samples. So here it talks about analyzing animal feed samples, cereals, milled, and all these kind of different samples. So when you are analyzing all these different samples, so what is the importance of calibration? So how your instrument needs to be calibrated here, the reference method. So the data that you feed in from the reference method or the primary method, how it shall be collected and how it should be put in the software. It talks about that. It also talks about bias correction. Now again, this NIR being a secondary technique, it also allows this bias correction. Wherein what happens is if the difference between the primary, your actual reading and the desired reading, it persists. So you could add a bias there. So the aggregate could be added there with a plus or minus thing, and then the differences or the additions would be made automatically by the instrument, and the readings would be given to you. Next is this expansion of calibration set. Wow, now this is another feature that this ISO talks about. So in what happens, suppose you have a calibration, a ready prepared calibration for your samples, but you've upgraded your sample to some by adding few more steps or so. Now, older calibration is no longer giving you exact or good results for your updated. But then this ISO 12099 also tells you like how you can expand an existing calibration with more data points, giving you reproducible, reliable results every time you analyze your samples. Next, it also talks about the statistics, performance, and measurement. So, the statistical analysis that the instrument performs to give you the results and achieve those results every time you analyze your sample, it also tells about that. So, this is what our ISO 12099 talks about. And here is where Unity Scientific plays a role. Like, why do you need Unity Scientific? Because it is trustworthy, easy to operate, easy to use, doesn't require any maintenance, So the calibration has to be performed at your own end only. The sample doesn't need any sample preparation. The instrument doesn't require any chemicals for its functioning, and all these things they contribute to this little maintenance. Next is it uses this besting town technology, so wherein it uses this monochromata based technology for bit of different cells. Next is this environment friendly, so as it doesn't consume any chemicals, it doesn't generate any waste. Okay, and as the same sample could be used for some other kind of analysis as well. So eventually, the waste and the that is hazardous waste and the less hazardous waste, and nothing is being generated out of it. So as no waste is generated, no time, no chemicals are being consumed and those things, it elevates your company's bottom line. You save your costs here, and eventually you get to a better line wherein just one single instrument is able to uh, do work of your 10 analysts and give you better results than them. Okay, so our instrument is trusted by PepsiCo, Mondelez, Nestle, Kellogg's, and many more around the globe. So even you could trust us for your products, for your attaining your batch to batch consistency, for attaining customer satisfaction, and eventually, when your batch is consistent, your customers are happy, you make good profits, right? So that is where Unity Scientific would be helpful to you. Thank you.